If you want to cure an ailment, you usually have to choose your venue. You either visit the local healer, grinding fresh leaves in a wooden mortar, or you trust the sterile, humming centrifuge of a modern clinical laboratory. We tend to categorize our health into two rigid buckets. We assume synthetic drugs are effective and natural plant remedies are safe. In reality, the line separating organic folklore from synthetic pharmacology is often invisible. The Philippines systematically dismantled that divide in 1997 with the Traditional and Alternative Medicine Act, or TAMA. The government realized that indigenous societies held generations of vital medical data that was being ignored by modern healthcare systems. To bridge the gap, the Department of Health established a clear objective. They took native Philippine flora, plants used for centuries in rural villages, and ran them through the same phase 1, 2, and 3 clinical trials required for any synthetic chemical drug on the market. Clinical validation transforms these common backyard plants into multi-targeted biochemical systems, providing the same level of pharmacological certainty as any modern prescription. The secret to these botanical treatments lies in a pharmacological principle called synergy. Synergy occurs when a combination of substances produces a therapeutic effect much greater than the simple sum of its parts. 1 + 1 = 3. Standard pharmaceutical practice typically isolates a simple active chemical from a plant, like extracting pure salicin from willow bark to make aspirin, and discards the rest. The traditional approach, however, utilizes the entire plant extract. A whole plant extract deploys a rich matrix of phytochemicals, attacking a disease across multiple targets. While primary compounds hit the main receptor, secondary molecules, like flavonoids, facilitate transport across cell membranes, lower parasite resistance, and protect the gastrointestinal tract. By stripping a plant down to a single, isolated chemical, modern manufacturing often removes the natural safety buffers and synergistic boosts that the plant evolved over millions of years. You can see this whole plant efficacy clearly in lagundi, a 5-leaf shrub that Filipino ancestors historically boiled into a tea to treat harsh coughs and colds. Modern clinical trials reveal exactly how it works. Lagundi acts as a highly potent bronchodilator by inhibiting the 5-lipoxygenase pathway. That pathway is the specific enzyme chain responsible for triggering inflammation in asthma. By blocking it, the plant extract forces constricted airways open, relaxing the smooth muscle and restoring normal breathing. We see similar precision in sambong, an aromatic herb documented for decades in local ethnological literature as a traditional diuretic. This chart compares sambong leaf extract against standard synthetic thiazide diuretics. Phase 3 trials demonstrate that sambong increases urine volume to flush out kidney stones at a rate comparable to the synthetic drug. However, while the synthetic thiazide depletes the body's essential potassium levels, sambong leaves the patient's potassium entirely intact. The folk remedies passed down through oral tradition were precise, targeted biochemical interventions. It simply took clinical science a few centuries to build the tools necessary to measure them. That same clinical rigor applies to external dermatological treatments, specifically indigenous plants like Acapulco and native bawang or garlic. When treating severe cutaneous fungal infections, native garlic relies on agewein. Agewein is a powerful sulfur-containing compound that forms only when fresh garlic is crushed, and its mechanism of action is incredibly violent. Scanning electron microscopy data captures the exact moment of impact. Exposed to a joni, the thick, protective fungal cell walls violently deflate, completely collapsing into flat, lifeless ribbons as their internal cytoplasm leaks out. The garlic compound specifically infiltrates the cellular membrane to destroy the pathogen's mitochondria and storage vacuoles, neutralizing the infection while leaving the host's surrounding cellular structure unharmed. These compounds have evolved into aggressive antimicrobial systems. They provide a high-velocity attack on pathogens that matches the standards of modern clinical care. Identifying these powerful botanical mechanisms is only the first step. Translating them into a safe, reliable format for the general public requires a strict regulatory framework. Before they enter a laboratory, these remedies originate in bustling, unregulated local markets, sold in raw bundles based purely on historical usage and local trust. When these plants are first processed, they often enter the market under the baseline tier as herbal or food supplements. To get this label, manufacturers only need to prove the product is safe to consume. Because of this, the government mandates a strict disclaimer on the bottle, warning consumers that the product is not a registered medicine. This checklist illustrates the ultimate regulatory tier, true herbal medicines. These formulations demand specific therapeutic claims backed by Phase 3 human trials, acute LD50 testing to determine toxicity limits, and the Ames test to ensure the compound won't cause DNA mutations. True herbal medicine is a triumph of careful formulation. Indigenous knowledge provides the initial map, while the standard clinical verification process ensures every dose is both safe and effective.